Good morning, good afternoon, good evening, and for some of you, good night, I guess. And thank you for tuning in from so many different places to our webinar series, The New Tools of the Trade in Life Sciences, Technology that Enables Breakthroughs. And today's webinar topic is Optogenetics for Cell Death Research, Tools and Applications. My name is Renate Weitzbauer. I'm one of the leads of the webinar program here at Bioprotocol, an open access online journal with a mission to make research more transparent and reproducible. And before we begin with the webinar today, let me share a couple of quick announcements. So if you have a question, you can post them in the Q&A or in the chat section throughout the talk, and Katarina will address them at the end. And the webinar will also be recorded and posted on our website later, so you can revisit it at a later point. All right, it is now my very distinct pleasure to introduce the speaker of today's webinar, Dr. Katarina Shakarina, who is currently a Swiss National Science Foundation-funded postdoctoral fellow in the laboratory of Dr. Eike Latz at the Institute of Innate Immunity at the University Hospital in Bonn in Germany. She received a PhD in immunology from the University of Lausanne in Switzerland under the supervision of Dr. Peter Pross, where she developed multiple optogenetic tools that can be used to trigger the three most studied forms of cell death: apoptosis, pyroptosis, and necroptosis, with the aim to apply them to reveal the different fates of dying cells and the bystander cell responses to it in epithelial tissues. And in the lab of Dr. Katz, Katarina continues to combine her expertise in optogenetics and high-resolution microscopy with various biochemical and immunological techniques to further understand the complex interplay between cell death, inflammation, and tissue repair. Her work on developing these optogenetic tools has been published in the Journal of Cell Biology, and an extended protocol on how to use them can also be found here at the Bioprotocol. And with that, Katarina, thank you so much for being here, and the floor is all yours.
Thank you very much for the introduction, and so I will share my slides with you. Yeah, so today we are going to talk about optogenetics. Can you hear me actually? Perfect.
Yes. Yes, we can.
And but before we start, I would like to first explain why am I interested in this. So my scientific journey started in Ukraine, where I did my bachelor and master studies. And at that point of time, I also started to work in the lab. And my initial training was in cell biology and cell signaling and cancer and stroma cell interactions. So, and this basically got me initially interested in microscopy as well as in cell biology. And so after finishing my studies, I started my PhD in Switzerland in the laboratory of Professor Peter Brosch. And there I actually got even more interested in cell biology and also optogenetics. And during my PhD, I developed different tools for triggering different forms of cell death and then looking at bystander cell responses. So at the moment, as Rene kindly mentioned, I'm a postdoc in Germany, in Bonn, in the laboratory of Professor Eike Latz, and my current research focus is still cell death, particularly inflammasome pathway, and I'm continuing to look also at bystander cell responses in tissue homeostasis and inflammation. So, and today I'm going to first quickly introduce you to cell death, and then I will explain how we actually build optogenetic tools for cell death research as well, and like why to start with do we need this kind of tools, and how do you approach this. Then I will show you some applications of using this kind of optogenetic tools for cell death induction in various contexts, and then we will also discuss considerations and limitations of using optogenetics for your experiments. So I would like to start with cell death. So why cell death? Why is it interesting? If you look in the literature 100 years ago, people already observed that in embryos or even in the adult tissue, cells die, but they actually didn't consider that it's worth the attention, because if you think philosophically, every living thing dies, so it's not even surprising that cells have to die. Later, yeah. However, now we are really interested in it as a field. Why? It is estimated that the adult human body contains around 37.2 trillion of cells, which is really a lot. I don't have to convince you of this. And even more surprising, it's also estimated that homeostatic cell turnover, which means death of the cells and their recycling by neighboring cells, is around 50 to 70 billion cells per day. And so cell death in humans plays a lot of very important physiological roles, such as quality control to remove cells which are senescent or, for example, damaged. Also, it plays a huge role in tissue adaptation during embryonic development as well as in our adult life. And it's also a key component in innate and adaptive immunity. However, it is also known that there are a lot of pathological roles of cell death, and it is extremely important in autoimmunity as well as hyperinflammation, during infection, also during a lot of tissue degeneration and related diseases such as Parkinson's and Alzheimer's. They all involve different forms of cell death happening. And cell death also is a main driver of various forms of tissue injury as well as cancer. So if you look in the literature for 10 to 20 years ago, this is what people used to think. They used to think that there is only two forms of cell death, which is apoptosis, which they used to consider the only true regulated form of cell death. And also there is this non-regulated accidental cell death, which is called necrosis. And apoptosis is the one cell death which accounts for all the normal homeostasis, and necrosis is cell death which just happens by accident when cell is damaged. However, of course, this picture changed quite a lot in the past 20 years, and for now, we distinguish more than 20 forms of cell death. And here are some of those which were known in 2018, and we continue to add new and new forms of cell death to this list. And of course, all those different forms of cell death, they happen in the different contexts, and they also have different consequences. So for the sake of today's presentation, I will focus only on three of those, which is apoptosis. Again, this is the most studied type of cell death. It's caspase dependent and non-lytic, and also two forms of programmed necrosis, which people usually are a bit less familiar with, which is necroptosis. It's the only form of the kinase dependent cell death, as well as pyroptosis, which is another caspase dependent cell death, but it also happens with membrane permeabilization by the proteins called discernins. So if you look at those three forms of cell death, of course, one thing which we already can see from this slide is they differ in their molecular mechanisms, but they also differ in their outcomes. So of course, when this kind of cell death happens in tissue, those cells will produce different signals, which will be sensed by their viable neighbors. And those neighbors will also respond. They will induce different forms of functional adaptation, which will then result in tissue level and host level responses. The big question which interests me in my research is, what are the signaling and functional outcomes of these many forms of cell death? So, and in my research, I address this question using optogenetics. So why? As a cell biologist, I like to imagine my cell death pathway simple. For example, I want to think that we can really separate apoptosis, necroptosis, and pyroptosis into these very, very separated and defined mechanisms where you have this one pathway and then another pathway and another, and you can just activate them and compare. However, if you look at more detail at the architecture of those pathways, it's not exactly like this. We know that, of course, signaling pathways have very complicated nature with a lot of different protein-protein interactions, also a lot of different other signaling pathways feeding in this. Also, we know that there is a lot of crosstalk and interconnectivity between different cell death pathways. And what is even more, signals which we use to induce different forms of cell death, they can also act on other pathways. And so the solution which people have been using in the field for over 10 years is to develop this artificial clean systems where you basically don't trigger the pathway from the most upstream receptor, but you can actually isolate selected protein or basically selected node of the pathway and then specifically activate it. And this way you can actually reduce crosstalk between pathways. You can also induce engagement of other regulatory interactions, and also allows you to actually bypass all these pleiotropic effects of triggers which we commonly use, such as pathogens or cytokines and so on. So if you want to build a tool, this kind of clean tools for cell death induction, where do you start? So for our advantage is actually that there are common features in cell death signaling, and one of those is the modular protein architecture, which is common to different proteins in cell death pathways. What I mean by this modular architecture, that you can actually see that different proteins, which are involved in different forms of cell death, they all have these common elements or domains, which, for example, what you see here, for example, are CAR domains, which are present both in several innate immune regulators, as well as here you can see CAR domains in inflammasome pathway, or like dead domains also, and so on. And those domains are more or less independent, and you can actually use mix and match approach to replace them and then create new protein interactions. Another common feature in cell death signaling is there is very strong involvement of homotypic protein-protein interactions, such as homodimerization or homooligomerization in cell death signaling. What we often see is assembly of these multimeric complexes, such as apoptosomes or inflammasomes, which contain many copies of the same protein. Also, for example, for inflammasome, this is ASC filament. For those who are not familiar with the pathway, ASC is the protein which forms the core of inflammasome, and it's extremely important for the signaling. And it's really, this ASC filament is formed by really polymerization of this many copies of the same protein. Also the same you can see on the effector level, for example, for German pores and even caspases, which are the main executors of apoptosis and paroptosis. They also require dimerization or multimerization for their activation. So fortunately for us, when we see this, what's coming from a bit of synthetic biology side, what we can think immediately about is that you can actually manipulate these protein-protein interactions, and you can just drive this dimerization or multimerization to then activate those pathways. If you look at synthetic biology toolbox, there are two types of tools which can be used in this context. There are chemogenetic tools and optogenetic tools. So when we talk about chemogenetics, we talk about artificial protein domains, or like sometimes it's also endogenous domains of endogenous proteins which are present in other organisms, or also mutated domains, which can be dimerized or otherwise multimerized using chemical dimerizers. So chemical dimerizers are small molecules which will diffuse into the cell and then bind to those domains and bring them together. So the idea is that then if you fuse your protein of interest to those chemically inducible dimerizable domains, then you can also dimerize your protein of interest when you add your chemically inducible dimerizers. So another approach is optogenetics. Optogenetics, again, it enables dimerization or activation of your protein of interest, but this time not using chemicals, but using light. So in this case, your dimerizable domain is the photoreceptor, which is typically derived from plant or algae, and it can be activated with the light, then triggering activation also of your protein of interest. So, and again, going back to hemogenetics, there is really large amount of tools which have been developed mostly for apoptosis induction, but more recently also for paroptosis and necroptosis. And I'm not going to go into the details, but together with my PhD boss, Peter Brosch, we recently wrote a review where we summarize what kind of tools is available for chemically inducible activation of different forms of cell death. I want to focus on optogenetics. When I say that I'm doing optogenetics, this is what people typically imagine. However, yeah, and this is optogenetics, which is very well known as in neurobiology as neuronal or opsin optogenetics, and it involves activation of specific channels or GPCRs in the cell membranes using light. However, besides this neuronal optogenetics, there is a whole field of so-called non-neuronal optogenetics, which doesn't involve opsins and doesn't involve, in the most cases, any kind of membrane channels, but those are all the different types of photoreceptors, also light-activated enzymes, which can be expressed in the cytoplasm or specific compartment of the cells and then drive different types of protein-protein interactions or signaling. And so why to use optogenetics? Compared to other methods, optogenetics offers several very significant advantages. First, it's really superior spatiotemporal resolution because you can really activate your protein in milliseconds to seconds. You can also use light patterning, which I will show you in a minute. Also, you can achieve ligand-independent protein activation because in this context, your ligand is light. And the advantage of this is that you don't need to add anything in cell culture medium. You don't need to worry about diffusion kinetics. You don't need to worry that your cells might pump out your compound. It's also relatively easy and, as I already said, fast, given if you have proper optogenetic hardware, which we'll also talk about later. And it also offers reversibility and possibility of bidirectional control. And so just to show you how efficient it is, this is actually a movie which I took from one of the published papers. So this is a system called improved light inducible dimer. And in this case, this is a heterodimerization system, which means that one of the partners is expressed in the cell membrane and tagged in green. Another one is expressed in cytoplasm and tagged in red. And when the light is turned on, they're going to come together. And so this is how it typically looks in the cell. So this is one of the examples how you can pattern the light in your cell to recruit your proteins to the very specific locations. So basically, again, what you see here, that where now you see these red shapes in shapes of the letters, this is where the cell is illuminated and all the protein which is tagged in red is specifically recruited to this particular location. So we're not using this system, we're using actually another system in our lab, and it's called Cryptochrome 2. And Cryptochrome 2 comes from plants, actually from Arabidopsis, which is one of the most used plants, to my knowledge, in the whole plant biology field, and probably one of the most studied. And we don't also use a full protein, we only use a separate light-responsive domain. And also this domain also contains additional mutation, which was introduced to enhance its homooligomerization properties. And what this protein does is that it senses blue light, and then it responds by forming large homooligomeric clusters, which have tens to hundreds of copies of the protein. And it's extremely fast with activation of kinetics of 15 to 75 milliseconds. And then after you turn off the light, it can also be reversible, so those complexes will actually fall apart. To show how it looks in the cell, this is one of our cells expressing just Cryptochrome 2 olig M cherry. So you can see that in the absence of activation, it just diffuses localized to cytoplasm. So we illuminate the cell with a single pulse of blue light at 5 seconds, and you can see what happens. So this is basically, you see that all the protein which was before diffused in the cytoplasm, it really quickly comes together, forms those complexes, but as soon as you remove the light, those complexes actually start to fall apart. So how do we take this protein, and how do we build optogenetic tools for cell death induction? Our favorite type of cell death is paroptosis, and paroptosis is actually a type of necrotic cell death. It is extremely important in the context of infection, but also in the context of different autoinflammatory disorders. So basically, what activates paroptosis is different internal and external stressors. For example, UV illumination, your sunburn is actually also a form of paroptosis, but also sensing of different pathogens such as bacteria, viruses, even some parasites inside the cytoplasm of the cell. So all those signals will be sensed by different receptors, and they form this complex called inflammasome, which I briefly talked about before, which consists of really many copies both of receptor but also adapter called ASC. And the key protein in this pathway is caspase-1. Caspase-1 is the protease, which means that it can cleave another proteins, and in our case, it cleaves pore-forming protein called glycerin D, which will actually go then to the membrane and pinch these big holes and create these pores, and also different cytokines and other substrates. So when we look at caspase-1, again, what we can find that has this modular architecture, which I mentioned before. It has CAR domain, which is involved in upstream interactions, and also caspase-1 homodimerization. and effector protease domain. And in my PhD research, we followed a very simple approach. We focused on keeping effector protease domain, and then we just replaced the CAR domain, which is normally involved in interactions with the upstream activators of caspase-1, by this photoactuator domain containing CRY2 olig and mCherry that we can really nicely find expressing cells. And so basically what we get in this case, we get this optocaspase-1, as we like to call it. And when the cells are non-illuminated with the light, the caspase-1, this optocaspase-1 is inactive and it's present in cytosol in the diffuse state. So when we illuminate those cells with the blue light, this leads to clustering of CRY2 and of course also clustering of domains of caspase-1. And as I briefly mentioned to you before, this dimerization alone is already sufficient to activate caspase-1. So, which means that in this case, the protease domain can then interact and cleave with different substrates, such as, for example, glutamine D, IL-1, IL-18, and other cytokines, and trigger paroptosis and inflammation. And this is how it looks in the cell. So this is, again, cell expressing our optocaspase-1, chimeric fusion. And you can see here actually why we normally use mCherry to tag it, because you can really nicely see that those cells are expressing optocaspase-1, and there are two cells which don't have any protein. And you can see that when we start to illuminate those cells with the light, they immediately change their morphology. And the green and blue, which you see here, are actually reporters for this form of cell death. And what you also nicely see that cell death happens extremely quick, and also the neighboring cells which don't express caspase-1, they're completely not affected, although they do come and try to eat the cells which are dying. And while, as I said, paroptosis is our favorite type of cell death, we are also interested to expand this approach to control two other types of cell death, apoptosis and necroptosis, using, again, similar dimerization or oligomerization principle. And actually, one thing which I should also mention here, that so why do we know that dimerization or oligomerization is sufficient to activate those proteins? It's basically all from previous studies and also from the previous work which used chemically inducible dimerization, oligomerization. And so just to show you how it works, so on the left you have cells which were expressing optocaspase-9, on the right you have cells which express optimal KL, and they both undergo very rapid cell death after elimination, also displaying this very characteristic morphology of the dying cells. And so one of the reasons why we like to use optogenetics is because it enables really extremely highly efficient and extremely synchronized induction of cell death. And just to demonstrate it, here there is another movie. So in this case, again, we illuminate cells not from the beginning, but after some time. So in the beginning, we image them for 20 minutes with the blue light off. Then we turn on the blue light and we monitor cell death by using drug, which is called drug 7. So drug 7 is membrane impermeable. It will only enter into the cells when cell death is induced. And what you can see here is, as long as there is no blue light, the cells are completely fine and happy. As soon as we turn on blue light, they get permeabilized. And as I already mentioned to you before, the cell death is extremely quick and synchronized. And so here you can also see that we can use this to compare, for example, different cell death effectors among themselves in terms of kinetics. And while some of those are more efficient than others, it's still always very synchronized. And one of the advantages of using optogenetics that you can also actually manipulate levels of cell death and as well as speed of cell death by manipulating illumination parameters such as light intensity or illumination duration. So this is one example. In this case, for example, we illuminate our cells with the light of different intensity, and you can see how percentage of dead cells changes accordingly. Also, like this is another example. In this case, we measure cell lysis, and again, we see that the more light intensity we give to the cells, the faster they die, and more of them also dies. And this is also another example how you can play with the light by, for example, using different light cycle numbers for a short period of time, or also different duration of illumination. And so I showed you some of the tools which we developed. And of course, besides a lot of other labs working on this, and there is a whole repertoire of optogenetic tools for different proteins in cell death pathways, and even different forms of cell death, such as ferroptosis, being developed. And so just to show you some other tools which are not yet published, but which we also have in our toolbox, is we also can go actually one step up above caspase-1, and we can directly induce nucleation of inflammasome complex. And so this is how it looks in the cells. This is TYD is the short name for the receptor domains, which we, again, in this context, oligomerize. And we also have other protein. You can see that you can really very rapidly trigger this complex assembly. And again, downstream of caspase-1, we have this pore-forming protein called gazurmin D, which I also mentioned before. And we can also just directly activate this protein with light using the photocleavable protein approach, which is shown here. So this is, of course, always work in progress, and we are also trying to further improve our light-activated cell death effectors. And what we actually trying to use is better photoactuators, which will have faster kinetics and also faster kinetics of deactivation. And we are trying to reduce dark state activity, which is an issue in a lot of optogenetics and cell death studies. And also we are working on trying to reduce endogenous regulation and non-desired protein-protein interactions. Another thing which we're trying to achieve is to generate AAV-compatible optogenetic cell death effectors, because the constructs which we have at the moment are actually quite big and they don't fit. And also we want to expand to other cell death-associated proteins and pathways in future. So this is regarding the tools, but of course, a big question which you probably have at the moment, what those tools actually useful for. So for this, I want to show you some of the applications. So a few years ago, our lab and others actually found that even if you induce cell death, the cells don't necessarily have to die. And sometimes they can still survive by engaging different membrane repair mechanisms, such as, for example, escort-mediated membrane shedding. And it turns out this is extremely easy to mimic with optogenetics, because a huge advantage of optogenetics, which you cannot really achieve with any other kind of technique, is that you can really turn on the light for a very, very short period of time, which means that you can activate only a very small fraction of the protein for very particular time duration. And what we can achieve using this is we can very transiently permeabilize our cells, which is shown, for example, here. So again, we use drug 7 to monitor membrane permeabilization. And you can see that indeed, if we stimulate our cells with one pulse of illumination, they become slightly permeabilized, but then they're actually able to recover and return to normal morphology. So just to be sure that what we see is not an artifact of cells that just not been sensitive to cell death, we can also stimulate them with several more pulses of light, and then we can see that they indeed succumb to paroptosis and become completely dead. Of course, I already have shown you a lot of movies where you can see cells dying, and this is another advantage of optogenetics, because it's actually really nicely compatible with lifestyle imaging and different forms of microscopy. And in a lot of cell death studies, people often struggle that if you want to look really close at your cell, you don't know when the cell is going to die and whether the cell is going to die at all, because cell death responses are rarely 100%, and you always have percentage of surviving cells in the population. When you use optogenetics, however, you know that the cell which is expressing your photoactuator or your optogenetically activatable cell effector is going to die. And then, which means that you don't need to image now hundreds of cells. You can really zoom really closely to your cell of interest and look what is happening. So in this case, again, you're looking at HeLa cells, which is dying by paroptosis, and you see a lot of events which are happening. So here the resolution is in the minutes, and we image those cells every 5 seconds, which you could even go with a higher frame rate. And you can see, for example, how plasma membrane changes, but also what is happening to the nuclei. And the orange dots here are lysosomes. You can also see how, for example, lysosomes stop moving and actually get permeabilized and lost during paroptosis. And in my PhD, we also used this approach to map early cellular events during different forms of necrotic cell death. We performed high-resolution single-cell analysis of those cellular changes with more than 20 fluorescently tagged proteins and dyes. And what it enabled us to achieve is to create this roadmap of necrotic cell death and also identify common features and this early event and medium events and then late events, which was not really possible to achieve before. Another very big advantage of optogenetics is that by activating a very specific node of the pathway, you can actually now not really study physiological. I'll start differently. One of the big advantages of optogenetics is that now you can actually flip your research questions upside up. So I'm coming from host-pathogen interaction field, and a lot of people in my field actually study how pathogens induce different forms of cell death. So they're infecting cells with bacteria or parasites, then they're trying to identify sensor, adapter, effector proteins, and then what kind of cell death is triggered. However, what is usually not really well known is how those different types of cell death affect pathogens in this context. So using optogenetics or hemogenetics, we can actually flip this question and ask, okay, if we induce cell death in infected cells independently of pathogen activity, what will now happen to pathogen? And just to show you that this is useful, in this case, you're looking at the human keratinocyte infected with the parasite Toxoplasma gondii. I think many people who have cats might be also familiar with this. And so this cell is not dying by infection. The parasite inside is also perfectly happy. But what if we start killing it? Look what happens. So those banana-shaped things here are parasites, which were sitting in the cytoplasm. And you can see that as soon as we induce cell death, those parasites actually start to escape. And if you focus here, then you can see that one of those parasites actually escapes and immediately reinfects neighboring cell. And now we can also start asking, okay, so how do those parasites sense that cell is dying? What mechanisms do they use for escape and also for reinfection of neighboring cells? Another type of experiments where optogenetics is extremely useful is if you want to look at how neighboring cells respond. Going back to earlier studies which were performed in homotypic cell population using triggers, for example, such as UV or infection agents. In this kind of context, it's usually very difficult to separate the response which was induced by cell death versus the response which was also induced by the cell death trigger, but on the cells which actually didn't fully engage the pathway. If we use optogenetics, then we can really very selectively trigger death of the single cells without affecting their neighbors. And then we get this, what we like to call clean system, because any kind of response which you will monitor in neighbors is not the consequence of your treatment, it's really consequence of those single cell death events, very similar to what happens in the tissues. So you can see here, also we can start tracking differences in those responses. So we have here 3 movies where we use the co-culture of sender cells, which express cell death effectors to induce paroptosis, necroptosis, or apoptosis, and the neighbors don't express any of those effectors, so they are not going to die, but they actually express calcium sensor GCAMP. What you can see that this response is very differential. You see that apoptotic cells actually indeed remain rather silent, as we expect, while paroptotic and necroptotic cells, I remind you, those are 2 forms of prognathic necrosis. They induce this huge calcium wave, which we nicely see here. and here. And we can also look, for example, more closely how neighbors behave. And again, in this case, you see epithelial co-culture where we have some center cells in red and the neighbors which have nuclei labeled with DNA disurrectin. And you can see that while in the control cell, the cells which only express our photoreceptor, they are happy. The paroptotic cells, they die, and indeed they also immediately get kicked out from the monolayer, and their neighbors actually come and actively push them out. So this is epithelial cells. You can also make our system more complicated, and we can also look at epithelial immune cell interactions in also co-culturing our sender cells with immune cells. And again, in this case, so the purple cells here, again, they express optocaspase-1, so they're going to die via paroptosis. And the neighboring cells, which are not labeled here, are actually human macrophages. And you can see that, again, when we induce optogenetically this form of necrotic cell death, it gets immediately sensed by neighboring immune cells, and they start to crowd around those dying cells and actually try to remove them. Again, what I have shown you before was all done in cell culture. However, we can also use optogenetics for cell death induction in vivo. And so this work was done in collaboration with Maria Leptin group in EMBL Heidelberg and her amazing postdoc, Eva. And what they did is they also helped us to bring our optogenetic tools into zebrafish. So here you see zebrafish larvae. In this case, also we use inducible heat shock inducible system, and you can see, so this is a normal larva which doesn't express yet proteins, and this is larva after heat shock which expresses a lot of red cells. This is again optogenetic cell death effectors tagged with M-cherry. And what you can see here again, when we select those cells which express either apoptotic, optogenetically activated effectors or paroptotic effectors here, and we can really see nicely how those cells die, but also what happens. And what you can also see here, for example, that paroptotic cells, they get indeed expulsed from these epithelial layers, and well, apoptotic cells actually get fragmented into the subapoptotic bodies and engulfed by their neighbors. So what do you need to consider if you now want to use similar approach for your research question? First, what you need to know is the activation mechanism of your protein of interest, which you want to control, and ensure that it's compatible with one of the available photoactivators. So going back to the approach which we used during my PhD research is we knew that our cell death effectors are activated by homooligomerization, and this would be sufficient to trigger cell death. This is why we used protein which induces homooligomerization. However, if, for example, you know that your protein requires heterodimerization or it requires cleavage, there are also other photoactuators with those modes of action which can be used for this. Another consideration is that optogenetics really nicely works when you want to focus on specific part of the pathway or specific you're interested in specific protein with little regard to the endogenous regulation. If you're really interested in, for example, studying how the cell death pathway is regulated normally under physiological conditions, then optogenetics can still be used as a tool, but probably not main tool, because a lot of those endogenous interactions will be missing. Also, optogenetics is perfectly suitable if you want to look at single cells or specific cell populations in vivo. Again, one of the big advantages that then you can really select those single cells using light patterning and target them and not their neighbors. However, if you want to do this kind of experiments, you also need to know that those tools are genetically encoded, which means that you need to be able to bring the constructs into the cells or into your organism of interest, which sometimes can be very complicated. And you also need to ensure that you have available hardware, such as the suitable microscope or LED illumination device to induce this type of experiments. So if you still decide that optogenetics is for you, where do you start? First, as I mentioned before, you need to choose right photoactuator, which again, as I mentioned before, you need to consider the mode of activation of your target protein, but you also need to consider cell or tissue or model organism which you want to bring it in, as well as cofactor availability. And the reason for this is because different photoactuators or photoreceptors utilize different cofactors, and some of those utilize the cofactors which are widely available in, for example, mammalian cells or plant cells, but others might not. And there are some photoreceptors which, for example, utilize ficocyanobulin. Those photoreceptors come from algae, and ficocyanobulin is not normally available in mammalian cells. There are ways to overcome those limitations by, for example, bringing cofactor either externally by injecting it into the animal or adding it into the cell culture medium. But again, here you run into some limitations, for example, whether it's going to be taken up by the cells. And another way to overcome this limitation is, of course, to bring the whole pathway for the synthesis. But again, this makes your life more complicated. So after you chose your photoactuator domain, then you need to design your optogenetic tool. What you need to consider in this context is the position of photoactuator relative to the effector domain. For some effector domains, it doesn't really matter. But for some proteins, for example, in our case, one of the kinases we were working with, it's really critical that N-terminus of this protein is free, because the N-terminus is binding to the plasma membrane and actually permeabilizing plasma membrane. So of course, in this case, we have to position photoactuator on the C-terminus of the protein. What I generally recommend is if you generate fusion protein and you replace one of the endogenous domains, try to place photoactuator at the same position, and maybe we will retain endogenous linker. If you don't know the preferred position for your photoactuator, then you can generate C-terminal and N-terminal fusion and also play with the linker length, try to find the most optimal conformation which will work for you. Again, for the linkers, I generally prefer to retain endogenous linkers, but you can also play with flexible linkers or rigid linkers to see, for example, whether your effector domains need to be in close proximity, whether maybe they need to be further from photoactuator, but this also has to be determined empirically. What you also need to think is tags, because sometimes you want to have fluorescent tag to see your construct in the cells, and sometimes you actually don't want to have fluorescent tag because you want to image your construct together with other fluorescently tagged proteins or dyes, and this is also a good thing to keep in mind. Also, you need to think how you want to express it and which promoter you're going to select. In our case, when we are dealing with highly cytotoxic proteins, which trigger cell death, and we prefer to use inducible promoters such as tetracycline inducible expression systems, but sometimes non-inducible promoters also work. And sometimes also, especially if you want to bring it into a specific cell type, also into organism. For example, you want to express it in neurons, so you want to express it in muscle cells, and you also need to think about tissue-specific promoters. Important consideration is also construct size, which is a problem if you deal with really big photoreceptors, because then the whole fusion protein might become actually quite big. In my case, the size of cryptochrome 2 is approximately 500 amino acids, and then some of my cell death effector domains are also few hundred amino acids, and then if I clone them into the lentiviral backbone, the whole concept becomes really bulky, and I always have to keep in mind, okay, which antibiotic resistance gene I can have, or like how packaging is going to work, and so on. And this is also really big problem, for example, for putting those constructs into adeno-associated virus vectors, which people commonly use for in vivo studies, because my optogenetic cell death effectors don't fit anymore. So now we need to think about maybe introducing different photoactuator domain instead of Cre2, which would be smaller. And as I mentioned before, another very big consideration is whether you want to retain or remove endogenous interaction or regulation sites or domains from your protein. Again, this very much depends on your question, because, for example, if you just want to induce cell death, then you probably don't want to have these interactions. But if you're looking what else is recruited to the complex, then maybe actually you want to retain them. So after you're done with the tool design, you usually perform initial testing and validation of the tool. For this, you need to establish readouts for the effector activity. So you need to know that if you stimulate your cells with the light, if your protein is active, that you have really efficient way to see it. So in our case, when we look at cell death, we can look at morphology, but we also have a lot of markers, for example, dyes, which only go into the dead cells. And this really enables us to determine whether our constructs work, how much cell death they induce, what is the kinetics, and so on. I also usually recommend to perform light dose titration, because even if you read the papers, people use different hardware. They also use different amount of, like, the different intensity of light for different types of research questions. What works for other people might not necessarily work well for you. Also, the optimal light dose also depends a lot on your protein expression level, on how long you want to stimulate, and a lot of other parameters. And you also need to include necessary controls. What we did initially when we were building our tools, we also included light insensitive and catalytically inactive mutants, because one thing which we wanted to see is that we don't trigger any changes in cell viability or cell behavior via illumination itself, or via just expression of those proteins, because overexpression of photoreceptors by itself can induce different types of cellular stress. And if you express proteins involved in endogenous cell death pathways, even in the inactive form, they still can affect other proteins by binding them or different types of interaction regulation. And what is extremely important is also to include phototoxicity controls, which I will talk about in a moment. And so at this stage, this is often repetitive. After you test your initial design, you often have to go back to your computer and actually think about, maybe you have to redesign your protein, which often comes down to, for example, changing position of domains, changing linker, maybe sometimes also changing photoreceptor. And then after you're done with the validation of your tools, then you need to think about what exactly would be the experiment which you want to perform, and then you fine-tune your setup accordingly. For example, you might need to express additional markers, or you need to change cell types, and so on. So one thing which I want to also talk about is the commonly used optogenetics hardware, because this is also the part where a lot of people get confused. So a lot of optogenetic experiments which I did during my PhD were all done on the standard confocal microscope. Also, sometimes people use multi-photon microscopes, such as 2-photon, even 3-photon. One thing which you need to consider in this case, if you want to use multi-photon microscopy, is whether your photoreceptor can be activated by the 2-photon illumination or multi-photon illumination, but this is usually can be found either in papers, so if this information is not available, you would have to test it. And it is also possible to perform optogenetics on epifluorescent microscopes using so-called digital mirror devices, which also enable you to project your activating light, which is in our case, for example, blue light, at the very specific region of your cell population. And besides this, for population-based studies, people use wide range of multi-well illumination devices. So we use this, the one which is shown here in the middle, which is called light plate apparatus, developed by Jeff Tabor lab. So this enables illumination of 24 well plates with different LEDs. But now, if you look, there are also other devices such as LITOS, developed by Oliver Per's lab in Bern, and this one is much more customable, because you can then just basically create any shape which you want and illuminate all types of cell culture vessels, 96 well plates, cell culture dishes, flasks, fax tubes, and whatever you want. So those two types of illumination devices are most commonly used, but now, especially if you want to perform in vivo studies, there are also more sophisticated setups. For example, there are a wide range of implantable optical fibers, which are used by people who do a lot of neuronal activation using neuronal optogenetics. Now there are also wireless LED chips the advantage of LED chips in this case, that you don't have this fiber sticking out of your model organism, but you can really nicely implant it and then activate it using computer or smartphone. And even more interestingly, there are now a lot of new developments in the area of this upconversion nanoparticles. The advantage of those nanoparticles is that they're not implanted, they can be just injected into your animal, and you can illuminate them with a near infrared light. And this will actually trigger emission of blue or green light. So it brings very big advantage, because one of the major limitations of optogenetics, which I didn't mention yet, is that you always depend on your light delivery. And people who work with optical methods also know that if you want to deliver light in the tissues, you're very limited by light penetrance, and it also depends on the wavelength. For example, blue light, which we are typically using for experiments, it doesn't penetrate so deep. You can penetrate few dozens of microns, but you cannot penetrate, for example, 500 microns. So it is much more easy with the far red or near infrared light, because this one would go much deeper in the tissues. So if you use those nanoparticles, you bypass this limitation, because you can then illuminate very deep, and then you will activate those nanoparticles, which would then emit, for example, blue light exactly where you want it. So lastly, I also want to talk about some of the common pitfalls when using optogenetics. And so one of the problems which I see quite frequently is inefficient cell death induction. And there are several reasons for this. One reason which often comes up is a poor construct expression or activation. Again, activation usually depends either on construct design or also on endogenous regulation. Sometimes it's also presence of inhibitory proteins. And for example, here below, you see that this is testing of our constructs in different cell lines. And you see that for all three types of cell death, we can see different response in different cell lines. And for pathologies, for example, we see that HeLa cells are poorly sensitive to activation of one of our constructs, while actually all the other cell types much more sensitive. For necroptosis, for example, we also see that one of the constructs doesn't perform well, and also it depends on cell type. For apoptosis, actually, two of the cell lines don't respond to one of the constructs at all. And the reason for this is usually, again, first, endogenous regulation, because if you look at the cell death pathways, there is really very high amount of regulation going on, which prevents induction of cell death under the conditions when cells shouldn't die. And if you want to induce those types of cell death, you need to actually go over this threshold, which is not always easy using optogenetics. And another common problem which happens is it might be that your cell type just doesn't have downstream effectors. Another common issue is also negative selection of expressing cells, and this happens especially when you use non-inducible promoters and expression systems. So I recommend going for inducible expression system as much as you can, and also even good inducible expression system, they can still give some amount of non-desired leaky expression. This is why we try to usually protect our non-activated cells from light at all times. And if you need to culture them, you also can do this under the light, which doesn't activate your construct. So for example, in our institute, when people work with optogenetics, we also have a room which is equipped with different LEDs. And if you know that your system is activated by blue light, you can still work under red light. And yeah, I already talked about negative endogenous regulation, low expression. And another very common issue is phototoxicity and photodamage. And the problem is that sometimes this effect is not obvious. And because if your cells die from phototoxicity, this is easy to see. But still, some amount of phototoxicity can also change behavior of your, let's say, protein of interest. It can also change regulation, because there are a lot of cell stress pathways which are activated in response to production of reactive oxygen species, and you might not see it via your immediate readout, but it can still affect your experiments quite a lot. This is why I also recommend always start with calculating light intensity and illumination duration before experiments. This can even done on the cells which don't express optogenetic proteins at all, to see whether it doesn't alter your process which you chose to study. And we also recommend, when you validate the optogenetic tool, to compare your response with the natural pathway triggers. For example, if you look at apoptosis, we typically just induce apoptosis using few of natural triggers and then see, okay, do we see same signaling? Do we see same cell death induction? Or do we see, for example, more with optogenetic because it's amplified by phototoxicity and so on? And yeah, so I also put some useful resources. If you want to search for the currently available photoactuators, then I highly recommend to go for Optobase. This is a database which contains all tools which people have built. It's constantly updated. It also contains links to the papers, so you can actually see how those were designed and how those were used. Also, if you want to get a very good general overview of currently available hemogenetic and optogenetic tools, specifically for cell death studies, as I mentioned before, me and my former boss recently wrote a review on this. Also, if you are interested in using some of the available tools for your cell death research, you can either look at our toolbox, which was published last year, and there is also a lot of work from other groups which you can find. And if you want to prepare your first optogenetic cell death experiment, then I really recommend our bioprotocol, which we published this year. And yeah, so of course, as I think all people know, science is not done by a single person. Doesn't matter how many authors are there on the paper. And I think I was, both during my PhD and postdoc, extremely lucky to work in amazing labs and being supported by amazing people. And so I want to thank, of course, my PhD advisor, Peter Brosch, and also the whole Brosch lab, where I developed most of those tools, and who actually also gave me freedom to go for this and try something which was originally considered just crazy idea, as well as my postdoc boss, who also fully supports me and gives me all the resources to continue and further develop my own ideas in this direction. And we also had amazing collaborators from EMBL Heidelberg who helped us with the zebrafish part and actually first time brought our tools in vivo, and of course facilities and all the different funders which were giving us money. And yeah, I would be very happy to answer questions. And if you have also further questions, you can contact me either by my email or also follow me on Twitter.
Thank you so much. This was a fantastic talk. Thank you also for outlining the design process in your last few slides, which I'm sure is pretty useful for everyone who's interested in setting up an optogenetics cell death experiment in their own lab. We have quite a few questions, so let's start with Asutosh Root. I hope I pronounced that right. How would we make sure that the kinetics and databasicity of binding of a ligand is recapitulated by light activity?
It's actually an excellent question. So most likely the kinetics will not be completely recapitulated, again, largely due whether, because like if you induce pathway naturally, usually starts from the most upstream part. If you use synthetic biology tools such as optogenetics or chemogenetics, usually go somewhere in the middle of the pathway and you trigger a very specific protein. However, if you want to, let's say, ensure that your activation, let's say you want to ensure that you induce the same level of cell death via optogenetics as you induce by your natural trigger, then I would just try to titrate expression level first, also the amount of light, until you get the numbers roughly similar.
Okay, excellent. And that leads us immediately to the next question. How can one determine the intensity or dose of light to be used? You said optogenetic induced cell death is light dose dependent.
Yes. So there are I don't remember the name of the device, but there are special devices which can measure light intensity. This is usually your imaging facility is equipped with some of those, because those are, for example, typically used to measure confocal laser intensity and to ensure that lasers work properly and don't change over time. And then you can actually quantify, for example, if you measure your laser intensity over a point of time over a certain area, like what is the dose? And yeah, I think, and I also heard from other people in the optogenetic field, that it's not really always necessary. What you want to ensure is that you know your microscope well. And initially, when I was doing also those experiments, we didn't start with a measurement, but we started with setting up laser intensity, let's say 5%, 2%, 1%, 0.5, and so on, and finding this laser intensity which worked the best, but didn't induce phototoxicity, and then just keeping this constant.
Thank you. That makes sense. Also within that realm of light sources and light intensity, one question is, which type of light sources are these effectors sensitive to?
So our toolbox is sensitive to blue light, which means that you can use LEDs from, let's say, 450 to roughly 500. And yeah, in my PhD, we had a LED device which was equipped with LEDs emitting 550 nanometer light. And also, when we work with confocal microscopy, we usually use 488 nanometer laser. But again, we also have some tools which are sensitive to red light, and there are also some tools which are sensitive to violet light. So then you just need to basically buy LEDs or be sure that your microscope can do this.
A really interesting question on using the different tools in the same cells. Or can you mix different cell death pathway optogenetic tools to observe them at the same time and in the same single cell? Have you tried that?
This is actually also really excellent question. I didn't try it, but we discussed this some of the time during my PhD, and it would be possible. The problem, or like the question, how to do it, of course, depends on your experimental setup and what exactly you want to see. You can theoretically express two different tools activated by the same type of light with different fluorescent tags. For example, you can use blue light sensitive tools, one type of tools tagged with M-cherry, another one tagged, for example, with a far red or near infrared fluorescent protein. And then you will see that your cell expresses, for example, either similar level or it expresses more of one, more of another. Then you can activate those tools simultaneously and see, for example, which one would be more potent under those conditions. Or you can also have two different types of tools which are sensitive to different types of light. For example, you can have tool which is sensitive to blue light versus tool which is activated by violet light. And then this would be actually my preferred approach to go to this kind of experiment, because then you can actually really decide when you activate the first one, when you activate the second one. And most importantly, you will have very nice control, because you can only activate one type of the tools and then see the very clean response, but not activate the second one.
Thank you. Another question is, can this technique be used for determining cell death of cancer cells, which is modulated by chemicals like drugs?
So I would say, yes. I would say if you want to screen for inhibitors, yes, this can be used, definitely. And there is actually a big advantage of using optogenetics for chemical screens, because it is known that sometimes if you add, for example, two chemicals or more, they can interact among each other, triggering more toxicity or also sometimes blocking the effects. In this case, you will need to bring only one compound, and you can also have very, like, since you induce cell death only by illumination, you can have very nice dose-dependent response. At the same time, if you want to determine which type of cell death is induced by your drug, then yeah, this is not really a good approach, because your drug would already induce something. And there's optogenetics, yeah.
I guess the next question is going a little bit in the same direction, but using the whole zebrafish model system. Is this technique useful to investigate the drug's inhibitory effects on induced zebrafish models?
Yes, I would say the only issue which we found during our in vivo experiments is that sometimes you need to ensure whether, or like, how to make your drug to penetrate into the fish, especially if you're dealing not with the tissues which are located close to the fish surface. For example, if you work with the skin, then I think it's easier. But if you work with the deep tissues, you find that some compounds don't really penetrate enough. And but yeah, this you would have to determine empirically.
And another question is, have you tried activating the cell death receptors on a given ROI? Because that might give you some insight on how much receptor activation is needed to have a cell death pathway to be activated.
Sorry, can you repeat it?
Oh, okay. Have you tried activating the cell death receptors on a given ROI, so region of interest, so within?
Yes, yes.
Because that might give you some insight on how much receptor activation is needed to have a cell death pathway to be activated.
Yeah, so we performed a lot of region of interest experiments for cell death induction in multicellular population. I think actually accidentally missed this data here. We also tried to activate receptors, for example, to induce inflammatory complex formation on the side of the cell, and then see how response propagates. So what we did see is that for many cell death receptors, one of the features of cell death pathways, which I didn't mention, is that there is also a lot of amplification of the signal, usually by using adapter proteins. So what we do see that it's sufficient to have very small amount of the activated receptor. As long as you have enough of adapter protein, then this will be enough to trigger the response.
Thank you so much. All right, I think this is actually it. Thank you so very much for an interesting talk, Katarina, and thank you everyone for being here. If you have more questions, as Katarina already mentioned, please feel free to reach out to her, or also to go on our webinar website where we have a Q&A section as well. And as I mentioned, this webinar is being recorded and will be posted on our website later, especially for the people who weren't able to attend today. And thank you again, everyone. Thank you so much, Katarina, for being here. That was really, really, really great.